Hi everybody, welcome back to Bio 155. It's been a little while. So our first lecture is going to start on how cells reproduce, which I've characterized on your academic plans as the M&M module, or mitosis and meiosis, and we'll be starting today with mitosis. But I'm going to first go through a lot of the nuts and bolts of how we're going to carry out the class for the rest of the semester, okay? So this is how we're going to organize our class in the post-coronavirus era. I'm going to do everything now, no exams, obviously. There's no way to proctor them and do them. So we're going to do everything by smaller organized units. So I'm breaking them down into what I call modules. The first module will be mitosis and meiosis, or how cells reproduce. I'm going to post on DropSend a set of the slides so that you can follow along with your own printout if you wanted to, or your own slides on your own computer to go along with these lectures that I'm posting on YouTube. You can take your notes down that way. I plan to include some lab information to sort of give us a lab component. You'll see what I mean by the time we get to the end of these slides. And then instead of having an exam, rather at the end of each module, I'm going to have kind of some problems or work set that you can turn into me. And as I've previously posted to you, there are several ways that you can turn in your material to me. You can turn it in as an attachment to email. You can fax it to me, and I've given you my fax number. As a last resort, you can send it in the mail to me, and I gave you my home address. And you can also post it up to DropSend if that's easiest for you, because sometimes you might need to post something that's a larger file, or you're scared of things not attaching to your email. Okay, and on DropSend, there is a section under the coronavirus. There's a folder called coronavirus announcements, and I think that's where I posted all the different ways to get a hold of me. So you obviously have my email address, but my fax number, my home address, my phone number, all that is in there. Now, instead of organizing this as an online class where you have to do it at a specific time, I'm choosing to do it by YouTube videos, so you can watch and rewatch videos, and you can answer your test questions or practice module questions along while you're watching the videos. And that way you can do it at your own pace. I know, especially now that everybody's not keeping a schedule, I've noticed with my A&P students, I get a lot of things at 3 o'clock in the morning, because a lot of you are at that age group where you're night owls. So this kind of accommodates to that. All right, so let's look a little bit at some DropSend things. All right, so some of you are still having problems with DropSend, and so I posted a video on how to both download information from DropSend and also how to upload. And most of you have already been downloading things from DropSend, so I'm not worried about that. But if you need to upload something to DropSend, here is the page, your first page, when you click into the storage folder, and you can see your regular folders, the A&P folder, the Bio 155 folder, the one where I was loading career information, the coronavirus announcements, general bio questions. I don't know if you've ever looked at that before, but there, I think, is some material on my mitosis and meiosis, because people always need to review that. The last folder is called Turned into Dr. H, and this is the folder where you can upload information to me. So if you have a file, like say you've answered all your test questions on the answer sheet, and you've typed it all in or written it all in, and you've scanned it into your computer. If you typed in your answers on the computer, you don't even need to scan it in. Okay, so what you can do then is take that file, and you see where it says folders, and there's icons going across the top there: add, edit, share, send, download. Okay, now up in the right hand corner, there's a arrow pointing up, and it's called online or storage. What you would do is click into the folder where you want to upload it and then hit that online storage button. So let's look at the next slide. The next slide shows where it says turned into Dr. H, and there is a Bio 155 turned in folder. You would then click on that Bio 155 turned in folder and then click on where it says the arrow online storage, and then you can select whatever you want to upload from your own computer and upload it and turn it into me, and I'll be checking those folders a couple of times every day. Now, several of you have work that you still need to turn in from the pre-coronavirus era, and I want you to turn that in as soon as you possibly can so you're not getting farther and farther behind. The last day of class, when we had the practical exam, we had about half of you show up. And I passed out take-home exam number 2 on that day. So if you were not there that day, you didn't get a hard copy of the exam. So you will need to download that. I've sent it to you already in email a couple of times, but it is posted on DropSend. And you can download that, do the exam. These are all lectures that we had prior to the coronavirus pandemic starting. We were supposed to take our lecture exam the following Tuesday, so that was the first thing you were supposed to be doing when we got back. So we've already had all the lectures for that. Okay, but we will review a little bit of what's going to be on that today. So please use the answer sheet that I provided with that. It just makes it so much easier for me to grade, and this is being a grading nightmare and trying to keep track of all your emails and your attachments for 50 people in 2 different classes is really difficult for me. So if you can try to stick to using the answer sheets, that would be really helpful. But whatever, hand it in as any way that you can, okay? Now, I've also posted all the grades, and it's updated for everything that I've had handed in to me so far. I've even updated it as recently as 5 o'clock this morning. So if there is something missing, I don't have it, and it is your responsibility to check through the grading file and see what I am missing for you. There are a few people who have absolutely nothing recorded for this class, and I'd almost recommend that you contact the administration and see if it's possible for you to do a late drop that just does not penalize you in any way. And maybe they'll just let you retake the class over when we get the classes going again. Okay? Other responsibility for you will be to check this YouTube channel regularly and DropSend regularly to see if there is anything new posted. I'm going to stick with the academic plan, and you all need to turn in that you agree to the academic plan and that you understand it. Okay? So let's get to it. Let's review a little bit about DNA before we get on to mitosis. We'll sweep out the cobwebs from our brains and see if you can remember what we've already learned. And hopefully this will help you a little bit for those of you who have not turned in exam 2 yet. And I can see already where some of you have some problems with some of the exam 2 material questions that a lot of people missed. This is a clinically oriented class, since most of you are going into health careers of some sort. So we, of course, are focusing our efforts of study on the eukaryotes, not the prokaryotes. The prokaryotes are cells that do not have a nucleus, such as bacteria. But you do probably want to know a little bit of what's going on there, especially because a number of you have to go on and take microbiology. So prokaryotes are different, remember, don't have a nucleus. All their DNA is out in the cytoplasm. Our eukaryotes have their DNA contained within a nucleus. And this slide reminds us of how our eukaryotes have a strategy of packing in or compressing all of that DNA, all that genetic material into their nucleus. So eukaryotes have chromosomes that consist of linear DNA molecules. And to pack it tightly into the nucleus, at the most basic level, our DNA is 2 strands of linear DNA wound in a double helix, and the 2 strands in that double helix are held together by hydrogen bonds. 3 hydrogen bonds between cytosine and guanine, and 2 between adenine and thymine. This is what you see at the smallest level at letter A. At letter B, we see the DNA wrapped around proteins known as histones to form a structure called a nucleosome. The histones are evolutionarily conserved proteins that are rich in basic amino acids and form an octamer. The DNA, which is negatively charged because of the phosphate group on it, is wrapped tightly around this histone core. The nucleosome is linked to the next nucleosome with the help of linker DNA, and this is the beads on a string model that you will hear about. At letter C in this picture, the DNA protein complex is called chromatin, wherein the mass of protein and nucleic acid is nearly equal. There's 2 types of chromatin in a cell. Heterochromatin appears as small, darkly staining, irregular patches that are scattered throughout the nucleus or accumulated adjacent to the nuclear envelope. Euchromatin is dispersed and not readily stainable. The euchromatin is prevalent in cells that are active in transcription of many of their genes, while heterochromatin is most abundant in cells that are less active or not active at all. This differentiation of the 2 types of chromatin is due to control over gene expression. Specifically, cells can control access to their DNA by modifying the structure of chromatin. Highly compacted chromatin simply isn't accessible to the enzymes involved in DNA transcription, replication, or repair. Thus, the regions of the chromatin where active transcription is taking place, called the euchromatin that we just described, is less condensed than the regions where the transcription is inactive, namely the heterochromatin. Then in D and E, you have some higher order levels of structure that are a little bit less well defined and less understood, but you have some loop domains that rest on some protein scaffolds, and you can have some high level expression of genes in the loops. At F, we see a depiction of the metaphase chromosome, a stage in mitosis, part of the cell cycle, in which chromosomes are at their second most condensed and coiled stage, the most condensed stage being that of anaphase. And this is what we will be discussing as we get into the cell cycle and mitosis. But first, let's review replication and transcription. Transcription and DNA replication both involve making copies of the DNA in a cell. Transcription copies the DNA into RNA so you can make protein, while replication makes another copy of the DNA so the cell can enter mitosis. Both processes involve the generation of a new molecule of nucleic acids, either DNA or RNA. However, the function of each process is very different, and you need to understand this. One, transcription, is involved in gene expression, and the other, replication, is involved in cell division. Although DNA and RNA have some structural or chemical similarities, each molecule performs different functions in living organisms. So transcription on the right-hand side involves copying DNA into RNA, and you will use the enzyme RNA polymerase to make a copy of a transcript of messenger RNA. This first step in the process is the unwinding and separation of the two strands of the DNA helix. The enzyme RNA polymerase then travels along the length of the strand of the DNA and binds complementary RNA nucleotides to it until a complete strand of messenger RNA is formed. The messenger RNA is essentially a cellular blueprint for the construction of a particular protein. Once the mRNA is made in the nucleus, it travels from the nucleus to the cytoplasm, where it is translated into a protein on the ribosomes out in the cytosol, either the ribosomes that are free in the cytosol or those that are associated with the rough endoplasmic reticulum. DNA replication, which is our primary concern for this lecture, is the process of copying the DNA in a cell so that there are two copies. This is done in preparation for cell division or mitosis. Before a cell divides, the DNA must be copied so that there's a copy for each of the resulting daughter cells. First, the DNA will be unwound and the two strands of the helix separate. An enzyme called DNA polymerase then travels along each strand, binding complementary nucleotides, the building blocks of DNA. And this will result in two double-stranded helices, which are an exact copy of each other. You will recall that DNA replication has three processes within it: initiation, elongation, and termination. So, as we just discussed, the eukaryotic DNA is bound to proteins known as histones to form the structures called nucleosomes, or the beads on a string. During initiation, the DNA is made accessible to the proteins and enzymes involved in the replication process. There are specific chromosomal locations called origins of replication where the replication begins. Certain proteins recognize and bind to the origin of replication and then allow other proteins necessary for DNA replication to bind to the same region. The first proteins that bind the DNA basically recruit other proteins. Two copies of an enzyme called helicase that you see here on the left-hand side are among the proteins that are recruited to the origin. Helicase unwinds and separates the DNA helix into two single-stranded DNA. As the DNA opens up, Y-shaped structures called replication forks, like the one depicted, are formed. Because two helicases bind, two replication forks are formed at the origin of replication. These are extended in both directions as replication proceeds, creating a replication bubble. Now here we only have one of the replication forks depicted. You can go back and look at our old notes to see the replication bubble. There are multiple origins of replication on the eukaryotic chromosome, which allow replication to occur simultaneously in hundreds to thousands of locations along each chromosome. Then we enter the elongation process. An enzyme called DNA polymerase adds DNA nucleotides to the 3' end of the newly synthesized polynucleotide strand. The template strand specifies which of the 4 DNA nucleotides, A, T, C, or G, is added at each position along the new chain. Only the nucleotide complementary to the template nucleotide at that position is added to the new strand. For example, when DNA polymerase meets up with an adenosine nucleotide on the template strand, it adds a thymidine to the 3' end of the newly synthesized strand, and then moves on to the next nucleotide on the template strand. This process continues until the DNA polymerase reaches the end of the template strand. Now, polymerase didn't initiate new strand synthesis. Primase was the enzyme that initiated polynucleotide synthesis by creating a short RNA polynucleotide strand complementary to the DNA strand. This short stretch of RNA nucleotides is called the primer. And once the RNA primer has been synthesized at the template DNA, primase exits, and then the DNA polymerase jumps on and extends the new strand with nucleotides that are complementary to the template DNA. Eventually, the RNA nucleotides in the primer are removed and replaced with DNA nucleotides. Once the DNA replication is finished, the daughter molecules are made entirely of continuous DNA nucleotides with no RNA portions. An extremely important point that you see depicted here in the slide is that DNA polymerase can only synthesize new strands in the 5' to 3' direction, adding new nucleotides onto the 3' end. Therefore, the two newly synthesized strands grow in opposite directions because the template strands at each replication fork are anti-parallel. So we have a leading strand that's synthesized continuously toward the replication fork. That's the one you see going continuously at the top in red, pointing with the arrow down. Okay? And helicase will unwind the template double-stranded DNA in front of that, getting rid of the supercoils so it's not twisting up. The lagging strand, which you see on the bottom half there, is synthesized in the direction away from the replication fork and away from the DNA helicase. This lagging strand is synthesized in short little pieces called Okazaki fragments. And this is because DNA polymerase can only synthesize in the 5' to 3' direction. So it constantly encounters the previously synthesized new strand. So now we're entering the termination phase of the DNA replication. The eukaryotic chromosomes had multiple origins of replication, which initiate replication almost simultaneously. Each origin of the replication forms a bubble of duplicated DNA on either side of the origin of the replication. Eventually, the leading strand of one replication bubble reaches the lagging strand of another bubble, and the lagging strand will reach the 5' end of the previous Okazaki fragment in the same bubble. DNA polymerase halts when it reaches a section of the DNA template that has already been replicated. However, DNA polymerase cannot catalyze the formation of the bond between the two segments of the new DNA strand, and it drops off. These unattached sections of the sugar phosphate backbone in the otherwise fully replicated DNA strand are called nicks. And we're going to need to put all these nicks together. And DNA ligase is the enzyme that ligates and connects at all of those nicks. After DNA ligase has connected all the nicks, the new strand is one long continuous DNA strand, and the daughter DNA molecule is complete. We then end up with our DNA replication that is semi-conservative, wherein each strand in the double helix is a template for a new complementary strand, and then we will have two separate DNA molecules, each having one original strand and one newly synthesized strand. And wow, that was an awful lot of information for one slide. So let's go to your textbook, which begins with the story of Ms. Henrietta Lacks. I'm happy to see that your textbook covers the story of Henrietta Lacks, whose story was untold for quite some time, and she has contributed immensely to the progress and advancement of medical science. Her story is, however, also a pretty sad story, in the sense that she contributed the first successful immortal human cell line, which was derived from cervical cancer cells that she unwittingly donated to medical researchers at Johns Hopkins University in Baltimore, Maryland. This was back in the 1950s, and things were a little bit more lax in the area of medical research ethics. As was the practice back then, Henrietta Lacks did not give consent for her cells to be cultured in a lab. And as it turns out, neither she nor her family was compensated for the extraction of her cells or their use. And her cells have been in long-term use in, as I said, advanced medical knowledge, and became basically a multi-million-dollar industry. Henrietta Lacks was an African-American tobacco farmer from Southern Virginia who got a bad case of cervical cancer when she was about 30 years old. A doctor at Johns Hopkins took a piece of her tumor without telling her and sent it down the hall to scientists who had been trying to grow tissues and cultures for decades without success. No one knows why, but her cells never died, so they became very useful, and they were passed around to researchers everywhere. Medical researchers have been trying for quite some time to grow human cells in culture, but the problem is that human cells tended to divide a limited number of times and die within weeks. And as I said, for some reason, Henrietta Lacks cells proved to be immortal and would just continue to keep growing in lab. So these were very, very useful. They divided very quickly in bottles of nutrient broth. So let's talk about Henrietta's immortal cells, famously called the HeLa cells, from her name, Henrietta Lacks. Medical researchers use her laboratory-grown HeLa cells to work and test theories about the causes and treatment of diseases. Her cell lines are immortal. They can grow indefinitely, be frozen for decades, and divided into different batches and shared among scientists. Her cervical cancer cells were the first immortal human cells ever grown in culture. They were essential to developing the polio vaccine. They went up in the first space missions to see what would happen to cells in zero gravity. Many scientific landmarks since then have used her cells for things like cloning or gene mapping and even in vitro fertilization. Now, Henrietta died a short time after her cells were harvested, and it was about 25 years after she died that a scientist discovered that many cell cultures thought to be from other tissue types, including breast and prostate cells, were in fact HeLa cells, because it turned out that HeLa cells could float around on the dust particles in the air and travel on unwashed hands and contaminate other cultures. Kind of sounds almost like coronavirus, doesn't it? And it became this enormous controversy because scientists experimenting on different types of cells had difficulty trying to figure out what genetic material came from the cells they were looking at, or possibly from HeLa cells. So in the midst of this, they decided to track down some of Henrietta's relatives to take some samples from them with the hopes that they could use the family's DNA to make a map of Henrietta's genes so they could tell which cell cultures were HeLa cells and which weren't, to begin straightening out all the lab contamination problems. So one day, a postdoc in a lab called up her husband, and he was not real sophisticated, only had a third-grade education. He didn't even know what a cell was. And so they make this phone call and say, hey, we've got these cells of your wife, and they're alive in a laboratory, and we've been doing research on her for the past 25 years. And now we kind of need to test your kids to see if they have cancer too. And this is probably not what the researcher actually said, but the scientist calling her didn't really know that the family didn't understand. And from that point on, all these problems ensued, and the family got sucked into the world of research, and the whole cell thing took over their lives. And her daughter, who didn't really know her mother because her mother died when the daughter was in infancy, the daughter's name was Deborah, somehow got involved. She always wanted to know who her mother was and was curious because she found out these cells were alive in the lab. And so she wanted to learn some information about it. She was basically hoping the cells could help her make some connection with her mother. But then Deborah's brothers discovered eventually that the HeLa cells not only were the first immortal cells, but they were the first human biological materials that were ever bought and sold. And with this being a multi-million-dollar industry, they became very angry when they found out that their family didn't get any of the money resulting from all this research. And that's pretty understandable, isn't it? They were a poor family. They had been intergenerationally poor, and they couldn't even afford health insurance for themselves. So they basically launched a campaign to try to get some of what they felt they were owed financially. And that's actually a question that continues to this day for all genetic research, is like, who owns a gene? And the idea of patenting DNA is an interesting legal issue. So Divine, maybe you can investigate that for us. But I think one of the key takeaway points here is to remember that we're dealing with human beings with feelings and connections and reverence for your own genetic material. And sometimes as scientists, we go down the track of always thinking of advancing science, and we forget about the very human nature of it all and the ethics. And I'm glad we pay a little bit more attention to ethics these days, because so much of science today revolves around using human biological tissue of some type. So hopefully we're now all more aware of the real human cost of all of this. So it is a very complicated story. It's complicated in that it's hopeful in all the wonders and great things for the advancement of medical science in solving perplexing problems of human disease and all the people that have been helped by all the research stemming from all of her cells. But it's also tragic in that she died and their family remained in poverty and that they were uninformed. And then they had a lot of bitter and angry feelings that arose out of this, and it's completely understandable that they would. I mean, how would you feel if you discovered your biological materials were being used without your permission? So these are fantastic research cells. HeLa cells are still used today. They divide very quickly in bottles of nutrient broth. The technology is still being used in research in biology, virology, medicine, every area of medical science. And it is hard to remember a time, I mean, it was before my lifetime, that scientists weren't able to establish tissue cultures with any reliability until they discovered these cells. So let's begin to look at the process of cellular multiplication and division with the backdrop of knowing So each of us started out as one cell, one fertilized egg. But by the time we were born, that single cell gave rise to about 1 trillion other cells, all organized as a human body. In an adult, billions of cells divide every day as we make new cells to replace our worn-out cells. Multicelled organisms, such as a human being, and free-living cells all have cell cycles. And biologists consider each cell in multicellular organisms to be individually alive, each with its own cell cycle. A cell's life passes through a series of recognizable intervals and events that are collectively called the cell cycle. Let's now examine the cell cycle. The cell cycle is also known as a cell division cycle, is the series of events that take place in a cell that cause it to divide into two daughter cells. These events include the duplication of its DNA, the DNA replication we discussed, along with the duplication of some of its organelles, and also the partitioning of its cytoplasm and other components into two daughter cells in an entire process called cell division. In eukaryotes, the cell cycle is divided into three main stages: interphase, mitosis, and cytokinesis. During interphase, the cell grows, accumulating nutrients needed for mitosis, and replicates its DNA and some of its organelles. During the mitotic phase, the replicated chromosomes, organelles, and cytoplasm begin to separate into two daughter cells. Cytokinesis is the finishing cell division, in which you have division of the cytoplasm and formation of a new plasma membrane around two individual daughter cells. Of course, all of this is highly regulated because it is so important that there are control mechanisms known as cell cycle checkpoints after each of the key steps of the cycle to determine if the cell can progress to the next phase. After each cell division, each of the new daughter cells that were formed begin the interphase of a new cycle. Think about it. From one fertilized egg, there are so many different cell divisions that a single cell develops into a mature organism. It is also the process by which we replace our hair, our skin, our blood cells, internal organs. All of our worn out cells are replaced by mitosis. And this slide shows you all of the divisions from interphase and then all the parts of mitosis. And we're going to go through each of those steps one by one. So let's start out with interphase. Typically, interphase lasts for at least 91% of the total time required for the cell cycle. Therefore, a typical body cell spends most of its life in interphase. Interphase consists of three stages during which a cell will increase in size, double the number of cytoplasmic components, and replicate its DNA. So let us look at the stages of interphase in great detail. There are three major stages of interphase: G1, S, and G2, and one side phase or quiescent phase called G0. They are named G or gap phases because outwardly they seem to be periods of inactivity, but they are not. A cell will start out its life in gap 1. During this phase, the cell roughly doubles the number of its cytoplasmic components, and it produces the molecules that it will need for DNA replication. Cells in G1 may temporarily or permanently exit the cell cycle by entering a reversible non-dividing state called G0. G0 is also called quiescence or a resting phase, where the cell has left the cycle and has stopped dividing. Non-proliferating or non-dividing cells in multicellular eukaryotes, such as a human being, generally enter this quiescent stage. They go into G0 after gap 1, and they may remain quiescent for long periods of time, possibly even indefinitely. For example, fully differentiated nerve cells or neurons enter G0, and they stay there. Because our nerve cells are terminally differentiated cells and are non-dividing, staying in this quiescent phase, this is why we have such a difficult time when we have neural injuries. As well, we have many cells that do not enter G0, and they continue to divide throughout an organism's lifetime, such as our skin cells, our epithelial cells. Now, of course, the cells that enter G0 aren't metabolically quiescent. They do go about their metabolic business. After the gap 1 phase, the S phase starts when DNA synthesis commences. And when DNA synthesis is complete, all of the chromosomes have been replicated. In other words, each chromosome will consist of 2 sister chromatids. So during this phase, the amount of DNA in the cell has doubled. The next phase is the gap 2 phase, which is a period of protein synthesis and rapid cell growth to prepare the cell for mitosis. During this phase, microtubules begin to reorganize to form a spindle before proceeding to the mitotic phase. The regulatory function, where cells must be checked at the G2 checkpoint for any DNA damage within the chromosomes, has to occur. This G2 checkpoint is mainly regulated by the tumor protein p53. We will learn about this later on, but I want you to know that it occurs here. If the DNA is damaged, p53 will either repair the DNA damage or it will trigger apoptosis of the cell. Do you remember what apoptosis is? Programmed cell death. When eukaryotic cells reproduce by dividing, each of its offspring receives DNA that's packaged in a nucleus. Mitosis is a nuclear division mechanism that maintains the chromosome number. Homologous pairs of chromosomes have the same length, shape, and genes. There is an exception, and the exception is for the sex chromosomes. The XY sex chromosomes in male are different. One comes from the father, the Y chromosome, and it is smaller than the X chromosome, which is inherited from the mother. Females inherit 2 X chromosomes, one from the father and one from the mother. So those are homologous chromosomes that do have the same length, shape, and genes. Okay, so let's discuss our homologous chromosomes versus our sister chromatids, because students always have a hard time keeping this straight. Okay, so here you see a picture of sister chromatids and homologous chromosomes. Homologous chromosomes are 2 physically different chromosomes that have the same genes but are not genetically identical. And you can look at these here where you have the blue homologue and the pink homologue and think that you inherited the blue homologue from dad and the pink one from mom. We could say this is chromosome 7. You inherited 1 chromosome 7 from dad, 1 chromosome 7 from mom. They are the same chromosome number and they have the same genes, but they aren't identical. Okay? Sister chromatids are 2 copies of the single chromosome, and they are genetically genetically identical. I'm tripping over my words. It's very hard to lecture when there's no one live. Okay, so they are genetically identical and they are physically attached to each other. So you started out with 1 blue chromatid and it then replicated. And so it is attached to its sister chromatid. Okay, do you understand that? Think about it for a little bit and make sure you get that straight in your head. Okay, now we're going to talk a little bit more about multiplication by division. A eukaryotic cell reproduces or multiplies by dividing. Sounds logical, right? When it divides, each of its two cellular offspring receives DNA packaged in a nucleus. The eukaryotic cell division necessarily includes a mechanism by which the nucleus divides. Nuclear division is mitosis, which maintains the chromosome number. Human beings are diploid, which means the nucleus of a human being contains two sets of chromosomes. As I said, one you inherited from mom and one you inherited from dad. Thus, all of your cells have two of each type of chromosome, except for the non-identical sex chromosome if you are a male. The chromosomes of each pair are called homologous chromosomes, and homologous chromosomes have the same length, shape, and genes. So here at A, you see what I was talking about on the previous slide. You have one chromosome that is blue from dad and one chromosome that is pink from mom. They are the same chromosome that I called chromosome 7, okay? Then, after DNA replication, when we look at picture B, we went through the S phase and each chromosome replicated. And now our chromosome from dad consists of two sister chromatids that are attached at the centromere. Likewise, our chromosome from mom, the pink one, now also replicated during the S phase and consists of two sister chromatids. So in B, we see that we have 4 chromatids, 2 sister chromatids from the chromosome of dad and 2 sister chromatids from the chromosomes of mom. Since they are replicated, they're ready to undergo division, which is what happens in C. And we see our sister chromatids separated, and one from mom and one from dad went into each cell. So we're back as we were at A, where we have one chromosome of each homologous pair inherited from each parent. Gene expression controls or determines when a cell divides. Some of these molecules regulating gene expression will cause the cell cycle to advance, and others will be brakes on the process. This control occurs at checkpoints built into the cell cycle, like the P53 that I was talking about earlier. Protein products of checkpoint genes interact to ensure that the cell's DNA has been copied completely and that it's not damaged, and even that enough nutrients are available to support cellular division. For example, a checkpoint that operates in S, where the cell is synthesizing new DNA, is monitored for damage during replication. Checkpoint gene products that function as sensors will recognize damaged DNA and bind to it. Upon binding, they will trigger other events that slow down DNA replication and enhance the expressions of genes involved in DNA repair. After the problem's been corrected, the brakes are lifted and the cell cycle proceeds normally. But if the problem could not be corrected, the gene products might initiate a series of events that will cause the cell to self-destruct, or apoptosis, programmed cell death. In multicellular organisms, mitosis and cytoplasmic division are the basis for the formation of the body and also increases in size during development. Continuing mitotic divisions of embryonic cells as they differentiate produces all of our tissues and body parts in the embryo. Juvenile development into the adult also occurs by mitosis, as does the ongoing replacement of damaged cells or dead cells. When you scrape your knee or pull out your hair, your cells are regenerated and continue to grow by mitosis. All of our growth and development and replacements of parts is asexual reproduction. We're reproducing new cells, but we're not rearranging the genetic material as when we make an embryo. So many plants and eukaryotes reproduce in this way. Now, our prokaryotes don't have a nucleus and they don't undergo mitosis. They will propagate by simple binary fission. So let's take a closer look at mitosis. Mitosis will proceed through four main stages: prophase, metaphase, anaphase, and telophase. And you really need to know these, so we'll go through them in great detail. A simple mnemonic device that I learned to remember the order is to remember pee mat. And think of the dog pees on the mat, pee mat. Gross, but it works. You won't forget it now. Now remember, before mitosis and its first stage, prophase, begin, the cell was first in interphase. And during this time, the chromosome. And now, since all that business was taken care of, in preparation for the nuclear division or mitosis, the chromosomes begin to condense and pack tightly. DNA replication and some transcription ends as the chromosomes condense into their most compacted X forms. And those X forms are, of course, the sister chromatids that are connected to each other. During prophase, the chromosomes are condensed that you can actually see them under a light microscope, which is what you would be doing in lab if we were having lab. Remember the centrioles that we learned about in, I think it was chapter 3 or chapter 4? Well, during prophase, the centrioles will replicate as well, and they have many tubulin subunits that are attached to them. So this whole region becomes what's known as a centrosome. The centrosomes will begin to migrate to opposite sides of the nucleus as the nuclear envelope breaks up. The microtubules from the centrosome will attach to the chromosomes. The lengthening microtubules form what's called a spindle, which is a temporary structure that moves the chromosomes during the nuclear division. The areas where the spindle originates on both sides of the cell are called the spindle poles. Let's look at a picture of early prophase. Here you see that the chromatin has been condensed into a highly ordered chromosome, and each chromosome is composed of two sister chromatids that are attached to each other at a central element of DNA called the centromere. When the chromosomes are paired up and attached, each individual chromosome in the pair is called a chromatid, and the whole unit is called a chromosome. When the chromatids separate, they will no longer be referred to as chromatids, but will be called chromosomes again. And this is where a lot of the confusion will come in for you. Okay, so the task of mitosis is to assure that one copy of each sister chromatid, and only one copy, gets into each daughter cell after the division. As prophase continues, the nuclear envelope will begin to disintegrate, and the lengthening spindle microtubules will penetrate the nuclear region and attach to the chromosomes at the centromeres. By the end of prophase, when we get into late prophase, one sister chromatid of each chromosome will become attached to a microtubule extending from one spindle pole. The other sister chromatid will become attached to a microtubule extending from the other spindle pole. The opposing sets of microtubules will then begin a tug of war. By adding or losing subunits, the microtubules will drag the chromosome toward the middle of the cell. When all the microtubules are the same length, the chromosomes become aligned midway between the spindle poles. And when they do this, we say we've now entered metaphase. And metaphase is where all the duplicated chromosomes line up midway between the spindle poles at what is called the metaphase plate. After metaphase, we enter anaphase. During anaphase, the sister chromatids detach and move toward opposite sides of the cell. When the sister chromatids separate, each becomes an individual unduplicated chromosome that we used to call a chromatid, but now that it's going to be in a new cell, it will be an individual chromosome. I hope you're not getting too confused by that. Then we go on to enter telophase. And telophase begins when one set of chromosomes arrives at each spindle pole and forms a cluster. Each cluster has the same number and kinds of chromosomes that the parent cell nucleus had, 2 of each type of chromosome. If the parent cell was diploid, you have 2 of each type of chromosome, and this is what occurs in human beings. A new nuclear envelope then begins to form around the clusters as the chromosomes loosen up. As the new nuclear envelopes start to form from fragments of the parental cell's nuclear envelopes and the chromatids become less coiled, at this point, telophase is ending and we enter the phase called cytokinesis. So let's look at pictures of all these 3 phases of metaphase, anaphase, and telophase separately. First, metaphase. At the end of prophase, kinetochores began to form at the centromeres. This is a complex structure that can be thought of as an eyelet for the microtubule rope that attaches. It's a point of attachment by which the chromosomes can be secured. The kinetochore is an enormously complex structure that isn't fully understood. 2 kinetochores form on each chromosome, 1 for each chromatid. And when the spindle grows to sufficient length, the microtubules find the kinetochores and attach to it. So at metaphase, the microtubules are attached to the kinetochores and they begin to line up in the middle of the cell. Proper segregation requires that every kinetochore is attached to a microtubule before the separation begins. It's thought that unattached kinetochores control the process, generating a signal, the mitotic spindle checkpoint, that tells the cell to wait before proceeding to anaphase. This is where everything lines up on what is called the metaphase plate, and the chromosomes are at their second most condensed and coiled stage. Metaphase plate is really important, and this is how you will identify cells that are in metaphase when you look at them under the microscope in lab. Now we move into anaphase. Anaphase is a stage of mitosis when the chromosomes separate and move to opposite poles of the cell. So these cells are easily identified under the microscope. Centromeres are broken and chromatids rip apart. When every kinetochore is attached to a microtubule and the chromosomes have lined up along the middle of the spindle, the cell proceeds to anaphase. This is divided into two phases. First, the proteins that bind the sister chromatids together are cloven, allowing them to separate. They are pulled apart by the microtubules toward the respective centrioles to which they are attached. Then, the spindle axis will elongate, dividing the centrioles and the set of chromosomes to which they are attached apart to opposite ends of the cell. These two stages are called early and late anaphase. At the end of anaphase, the cell has succeeded in separating identical copies of the genetic material into two distinct populations. They are at opposite poles of the cell. This is how you identify the cells in anaphase under the microscope when you're looking at it in lab. We now enter telophase. In telophase, the non-kinetochore microtubules elongate the cell and try to cut the cell in two. The nuclear envelopes start to become created by fragments from the parental cell nuclear envelope. Then the chromatids start to become less tightly coiled together. By this point, cytokinesis is fully underway. So let's look at a summary slide of mitosis before we go on to cytokinesis. And here you can see the pictures that we've gone through individually on the left-hand side for prophase, for interphase first, and then early and late prophase, and then metaphase, anaphase, and telophase. And then you can see on the right-hand side both processes happening in a plant cell and in an animal cell. And you can see they're readily identifiable stages, especially at the point that you get to anaphase. Look at that anaphase in the plant cell, the second one from the bottom, just pulling apart at the spindle. And you can go on to watch some really good YouTube videos that show the actual separation happening if you're interested. Okay, now it's time to discuss cytokinesis. And cytokinesis is the process of the cytoplasmic division that follows telophase. During cytokinesis, the amount of cytoplasm that goes into each new daughter cell is usually the same. In animal cells, the cell membrane forms a cleavage furrow and pinches apart like a balloon. In plant cells, a cell plate forms, which becomes the new cell wall separating the daughters. As we are more concerned with animal cells, let's discuss this further. In animal cells, there is a mesh of cytoskeletal elements underneath the plasma membrane. This is called the cell cortex. And the cell cortex reorganizes during anaphase to form a band of microfilaments and motor proteins that wrap around the cell. This band is called a contractile ring. It contracts after telophase, pulling the membrane inward. This is the cleavage furrow that partitions the cytoplasm. Let's look at a picture now of the cytoplasmic division. At the top, in this animal cell, you see a ring of microfilaments and motor proteins that form under the plasma membrane during anaphase. And this contractile ring wraps around the cell. After telophase, in the second picture, the contractile ring pulls the cell surface inward, forming the cleavage furrow as it constricts. The spindle microtubules guide the vesicles to the plane of cell division. And then, when the contractile ring is at its smallest diameter, the spindle microtubules inside of it are cut. Vesicles fuse to form new membrane that partitions the two new cells. For the plant cells, we aren't as concerned with this, but plants generally have a stiff cell wall around their plasma membrane, so the cytokinesis differs a little bit. Before mitosis, a ring of parallel microtubules will form around the future plane of division, and the microtubules form the spindle that by late anaphase have guided the vesicles to the plane of division. The vesicles fuse into a flat cell plate, which forms midway between the spindle poles. and this will partition the cytoplasm. The vesicle's contents are converted to cell walls. And here is a picture of that happening, but I will not test you on cytoplasmic division in plant cells, but it's here for you to look at. Now, an interesting concept that we need to talk about is the telomeres in cells. Telomeres are regions of repetitive nucleotide sequences at the end of each chromosome, like a cap that protects the end of the chromosomes from deterioration or from fusion with neighboring chromosomes. Telomeres shorten every time a cell divides, and when they get too short, the protective proteins can no longer bind to them, and the DNA at the ends of the chromosomes become exposed. Checkpoint gene products will then bind to the DNA and target it for repair. One of these gene products is P53, and it operates the checkpoint in G1 or gap 1. When a cell's DNA is damaged, this checkpoint moves the cell into the quiescent G0 phase until repair enzymes fix the damage. These enzymes can repair minor DNA damage, but they can't fix short telomeres, so the cell stays in that quiescent G0 phase. Cell cycle arrest that occurs because of telomere shortening tends to be irreversible. A cell in this state eventually dies or becomes senescent, permanently stuck in G0. It's metabolically active, but not functioning properly. And the result of this can be dangerous in the same way that it's dangerous for the accelerator of a car to get stuck, even when the brakes are working. A senescent cell becomes progressively abnormal. For example, enlarging and releasing too much of a protein that promotes inflammation. It will continue interacting dysfunctionally with other cells until the immune system detects it and eliminates it. Now remember Dolly, the first clone that was produced, Dolly the sheep, they attribute Dolly's short lifespan as being the result of short telomeres. Many have questioned whether telomeres are the secret to the fountain of youth. Most of our body cells can divide only a certain number of times before they enter senescence. And that number, or cell division limit, varies by species. Human cells can divide about 50 times before senescing. And that was one of the differences of the HeLa cells, is they didn't enter senescence. They were immortal. This pre-programmed cell division limit is thought to be protective in case a cell loses control over the cell cycle and begins to divide again and again uncontrollably. Limiting the number of divisions prevents rogue cells from overrunning the body. And when you think of it, the HeLa cells were rogue cells. They were cervical cancer cells. And I keep saying were, I should say are, because they are immortal, so they still exist. Telomere shortening is thought to be part of what sets an organism's lifespan. This is because as an animal ages, the number of senescent cells increases in its tissues. And when these cells accumulate, they disrupt tissue and organ function, a common problem with advancing age. So back to Dolly. Dolly's DNA came from a cell from an adult sheep that was about 6 years old. And when Dolly was 2 years old, her telomeres were as short as those of a 6-year-old sheep. Now, we do retain some pools of cell that have the ability to divide indefinitely or immortality. And these are called stem cells. And stem cells are immortal because they continue to make telomerase. Telomerase is an enzyme that lengthens telomeres by adding TTAGGG repeats to the end of the telomeres. These are those non-coding regions of DNA at the ends of the eukaryotic chromosome. Vertebrate telomeres consist of a short DNA sequence from the 5' end, TTAGGG, to the 3' end, and these are repeated perhaps thousands of times. This is that cap that protects the telomeres from potential damage. Both stem cells and cancer cells have high levels of telomerase, so perhaps telomerase is the secret to the fountain of youth. Again, telomeres shorten every time a cell divides. When the telomeres get too short, the checkpoint genes will stop the cell cycle and the cell dies or becomes senescent. Senescent cells are permanently stuck in gap naught, G naught. They are metabolically active cells, but they aren't functioning properly, and they become progressively abnormal. Most cells can only divide a certain number of times before they senesce, and this seems to set the lifespan of an organism. And in humans, it appears to be about 50 divisions. In an adult, only stem cells retain the ability to divide, and this is because they have the capacity to continue to make telomerase, the molecule that reverses telomere shortening that normally occurs after DNA replication. Again, cancer cells also have high levels of telomerase, and therefore they also divide indefinitely. Now let's talk about cell culture. Normal human somatic cells, those are your non-reproductive cells, in a culture plate have a limited dividing potential, just like normal human cells in the body. The telomeres will shorten with each subsequent division. And this was where the immortal HeLa cell became so vital and so significant to medical research. HeLa cells expressed telomerase, the enzyme that prevented the shortening. And let's look at that. Normally, when we put individual cells out on a primary culture plate, they will plate out in a single layer, as at the top of this picture, and they have contact inhibition with the edges of the plate. They then stop dividing, and a lab technician will transfer some cells to a new medium and start a secondary culture. Now, when we look at transformed cells in this bottom picture, transformed cells are another name for cancer cells usually, and we plate them out into a culture plate, petri dish. Rather than having the contact inhibition of normal cells, they will mound up on the culture plate. They are not inhibited in their growth. And this is also what the HeLa cells do. They are not affected at all, and they continue to grow regardless of density. And you can see how cancer cells do this, and it causes the formation of tumors in a body. When enough checkpoint mechanisms fail, cells lose control over the cell cycle. The cell and its descendants then form what's called a neoplasm. A neoplasm is an accumulation of abnormally dividing cells. Neoplasms that form a lump or abnormal mass in the body is called a tumor, and that's like that mounding plate on the culture plate. Cancer cells will grow and divide uncontrollably. They escape cell death. They are rogue cells. So you can see that cancer is essentially a disease of mitosis. When a cancer cell is transformed, all of the normal checkpoints regulating mitosis are ignored or overridden by the cancer cell. And I am going to end the lecture here because I'm having difficulty advancing the slides after reaching in over an hour of lecturing.